So here 5 to 10 defibrinated sheep or horse blood agar is added to a melted agar which is already melted at 100 degrees Celsius, and we are bringing down the temperature to 45 to 50, and then we are adding 5 to 10% defibrinated sheep or horse blood. So otherwise what happens if we are adding this blood to the 100 degree temperature agar, it gets lysed. So we should lower the temperature. And the blood acts as an enrichment material, and also it acts as an indicator. Enrichment means it provides many nutrients for the growing microorganisms in the media. Certain bacteria, when grow in the blood agar, they produce hemolysis. That means lysis of the blood or heme contents around the colonies. So we have seen the picture here. This is the bacteria microorganism which lies the surrounding media. So what happens? This blood content, with the help of the production of hemolysin, they lysed most of the blood content within the agar. That we are seeing the lysis here like this. Here also, and here, etc. Here a very small lysis is there. So it can be either alpha lysis. This is beta lysis, complete destruction. Here no lysis. That may be gamma lysis, okay. So we can explain the different types of changes which may occur in the blood agar. First is beta hemolysis. The colony is surrounded by a clear zone of complete hemolysis. Clear zone of complete hemolysis. That is this picture. Example is Streptococcus pyogenes. It is a beta hemolytic streptococci. Complete lysis means beta lysis. The next is alpha hemolysis. The colony is surrounded by a zone of greenish discoloration due to the formation of biliverdin. So here no complete destruction have been occurred, only a partial destruction. That is why a zone of greenish discoloration can be seen surrounding the colony because of the formation of biliverdin. Here I think this picture is not much clear. Actually, in the case of this alpha hemolysis, we can see a greenish zone of discoloration. Okay. The alpha hemolysis. An example for such bacteria is virulent streptococci, which can be seen in our mouth also, which causing plaque, etc. cavity in our teeth. Next is gamma hemolysis. Here no lysis have occurred. There is no change in the medium surrounding the colony. Look this colony. No changes. That means gamma lysis. Here also no change. Okay. So these are the three types of hemolysis. Next we are going to describe about chocolate agar or heated blood agar. It is a non-selective enriched growth medium. It is also enriched growth medium which is used for isolation of pathogenic bacteria. Here we are using chocolate agar for the isolation of pathogenic bacteria, and also it is a variant of blood agar containing RBC or red blood cells that have been lysed. Here in this medium, the RBC, we are adding blood similar to blood agar, but in this case RBC have been lysed. Why? Because we are adding this RBCs at a temperature 80 degrees Celsius. So that's why the RBC, while added blood when we are adding, it may cause lysis of RBCs. So the color of the media has been turned to brown color, which gives a chocolate color. That's why the name chocolate agar or heated blood agar. So here RBCs have been lysed by slowly heating to 80 degrees Celsius. Chocolate agar is used for growing fastidious respiratory bacteria. Those require specific nutrients. Such as, what are the respiratory bacteria which are fastidious? Haemophilus influenzae, and Neisseria meningitidis. These are the respiratory bacteria occupying respiratory tract, causing infections. In addition, some of these bacteria, most notably Haemophilus influenza itself, need growth factors such as NAD or nicotinamide adenine dinucleotide, that means factor 5, and hemine, that is factor 10, which are inside the RBCs. Thus, it is a prerequisite to growth for this bacteria. It is the presence of RBC cells lysates. The heat also inactivates enzymes which could otherwise degrade NAD. So this heat, as a result of this high temperature, it may inactivate enzymes also, and also which could otherwise degrade this NAD. The agar is named for its color and not because of the chocolate products which are present. So what happens in this condition is that here when blood is heated at high temperature, it may cause lysis of RBC, and these factors are released into the medium. So it may be a prerequisite for the growth of the bacteria, that means Haemophilus influenzae. Because these contents are released as a result while we are applying a high temperature. These contents are released. So this media may be useful for the Haemophilus influenzae. So this is the picture of uninoculated chocolate agar, and this is inoculated media. And the chocolate agar, with the addition of bacitracin, it becomes selective for the growth of genus Haemophilus. Another variant of this chocolate agar is called Thea Martin agar. It contains extra ingredients such as assortment of antibiotics. So it helps in the selective growth of Neisseria species, and because of the presence of antibiotics, it inhibits the growth of other unwanted bacteria. Only it may promote the growth of Neisseria species. That's why we are adding antibiotics to make that medium more selective. So it is a variant of chocolate agar. And here also in some chocolate agar we are inserting a disc of bacitracin. That is, with the addition of the bacitracin, it becomes more selective for Haemophilus. So in the laboratory, for the isolation of this Haemophilus species, we are adding certain bacitracin disc, antibiotic disc, into the chocolate agar after inoculation. Why? For the selective isolation of genus Haemophilus. Next is about McConkey agar. McConkey agar, it is an indicator and a selective differential culture medium for the bacteria, and it is designed to selectively isolate gram-negative and enteric bacilli, and it may differentiate them based on the lactose fermentation. So the McConkey agar, it is an indicator. It is selective, and it is mainly used for in the genus Enterobacteriaceae to isolate gram-negative bacilli and also enteric bacteria. That is, enteric means those found in the intestinal tract, and differentiate them, and we can differentiate the enteric bacteria based on the property of lactose fermentation or not. So the McConkey agar, which can be used to differentiate the bacteria whether it is a lactose fermenter or not. McConkey agar grows gram-negative bacteria, and those bacteria which appear differently based on their lactose fermenting ability, as well as the ratio of fermentation and the presence of a capsule or not. Most commonly used in the genus Enterobacteriaceae, and the ingredients of the McConkey agar is agar, peptone, sodium chloride, bile salt, lactose, and an indicator named neutral red. So it is a selective and indicator medium. Why we are saying selective medium? because it consists of bile salt. It does not inhibit the growth of Enterobacteriaceae because bile salt which are present in our intestine also. So we are providing the condition like that in the intestine for the growth of Enterobacteriaceae, but inhibits the growth of many other bacteria. So it helps in the growth of Enterobacteriaceae, this bile salt. So why this medium is telling as indicator medium? Because the colonies of the bacteria, if they ferment lactose, they takes a pink color because of the production of acid. And acid, as a result of the acid production by the fermentation, it turns the indicator present in the medium, which is neutral red, to pink in color. That is why the lactose fermenting colonies appear pink in color. Example for the lactose fermenters are E. coli. And we can also see non-lactose fermenters, those which doesn't ferment lactose. They appear as colorless colonies, and it indicates that the lactose is not fermented. That is, example for such bacterium are Salmonella, Shigella and Vibrio. So these are the examples for the plain McConkey agar, and this is an example for lactose fermenting colonies. Here the lactose fermenting colonies, here we can see pink color is the lactose fermenting colonies because of the change of the indicator neutral red to pink. Here no change in the color of the medium because no acid production takes place, so the indicator doesn't changes its color. So, and because of the no fermentation have taken place in this medium, this portion. The organism doesn't ferment lactose. That's why it appears as pale color. So here this is lactose fermenting colony. This is non-lactose fermenting colony. So differentiate to this both colonies we are using McConkey agar. Next is about Muller-Hinton agar. Muller-Hinton agar, it is a microbiological growth medium that is commonly used for the antibiotic sensitivity or susceptibility testing. So the Muller-Hinton agar or MHI, it is the most commonly used medium where we perform antibiotic susceptibility testing in our laboratory. And it is mainly used to isolate and maintain Neisseria and Moraxella species can be maintained in this media. And it typically contains 2 grams of beef extract. And we are performing disc diffusion method in the Muller-Hinton agar. A disc diffusion sensitivity test for antimicrobial drugs are carried out in this medium as per WHO recommendation to promote what? Reproducibility and comparability of results. The Muller-Hinton medium contain beef infusion, casamino acids and starch. The starch acts as a colloid in the medium that protect against toxic materials in the medium. So these are the ingredients of the Muller-Hinton agar. Beef infusion and casamino acids provide energy and nutrients for the bacteria. Agar we are adding as a solidifying agent. And the Muller-Hinton agar have few properties that make it excellent for antibiotic use. First of all, it is a non-selective medium and also non-differential medium. This means that all organisms can be plated on this type of medium. Starch is known to absorb toxins released from bacteria so that they cannot interfere with the antibiotics. Starch is known to absorb toxins. So they cannot interfere with the antibiotics. So starch is present in the medium. Then the Muller-Hinton agar is the best medium for the routine antibiotic susceptibility testing because of the following reason. It shows acceptable batch-to-batch reproducibility for the susceptibility testing. And also it supports satisfactory growth of most non-fastidious pathogens also. So it helps in the growth of both fastidious and non-fastidious pathogens. So these are the examples, picture of Muller-Hinton agar. Thank you.